that's the key point here. We are talking about something that is very, very fundamental to the cell. So I'm Max. I'm a group leader at the Francis Crick Institute, and our lab works in the interactions between macrophages and Mycobacterium tuberculosis, actually one of the most important pathogens for humans. All of the cells, all of the mammalian cells, will have a pathway where when they're under stress, they need to generate nutrients. So the way they do it is that sort of eating themselves, and this is like autophagy. Actually, I did my PhD in autophagy, and that was kind of a new idea. This was around 20 years ago, where we found that autophagy was part of the innate immune response, and cells were using this kind of fundamental biological process to destroy and attack intracellular pathogens. So there's been this idea for a while, but we weren't having really the right tools to study this in the context of human biology and in the context of human macrophage biology. In our recent paper, what we have done is we took human cells and we knock out specific genes that are for the autophagy pathway, because if you knock out those genes, now the bacteria can grow much faster and actually kill the cells. So it really shows you that this pathway is key for the control of Mycobacterium tuberculosis in human macrophages. So if you think that TB until, let's say, 15 years ago was a curable disease, but in the last decade and even longer than that, the emergence of drugs that are, the strains that are resistant to many antibiotics, and these are multi-drug resistant strains, is really going up. So finding strategies that allowed us to target bacteria that are resistant to antibiotics, for example, boosting our natural immune responses, it is important and relevant. And of course, autophagy in this context of infection has one function, but there are many other people looking at autophagy and targeting autophagy in other contexts like cancer and inflammation and many other diseases where autophagy is also very important.